Hello, everyone. We are Group 33. I am Group Lead Jermaine Duffus, joined by my teammates Alejandro, Elishiva, Arlie, and Kadeem. Our sponsor is Dr. Hank Zong, and our advising professor is Washim Ashgar, and our project is Multiclass Lung Cancer Classification. Lung cancer is within the top 3 most popular cancers by volume and is the deadliest cancer by volume. Despite this, lung cancer has a relatively low research and development rate. So our project is meant to address these issues by leveraging both classical and novel machine learning techniques to identify specific genetic biomarkers indicative of lung cancer. And our two main deliverables are a research paper that displays our findings for further peer review and research, and a web platform that allows non-technical users, such as physicians, to utilize the models employed within our research.
Approach. To ensure accurate lung cancer classification, we follow a structured approach. First, we pre-process and normalize high-throughput microRNA expression and methylation data, reducing batch effects and technical variability. Next, we apply VTFS and baseline models to select the most discriminative microRNAs. We then conduct a comparative biomarker analysis, integrating methylation markers to enhance classification robustness. These selected features are used in machine learning models, SVM, random forest, and SAINT, to classify cancer stage and subtype. Finally, we evaluate model performance based on accuracy, sensitivity, and specificity, ensuring clinical reliability across diverse datasets. Requirements and constraints. From a technical and medical perspective, our requirements were to use only publicly available plasma microRNA datasets, ensure that all our models are trained on de-identified data, classify across 3 tasks: diagnosis, stage, and subtype, and integrate feature selection to improve interpretability. Constraints included high dimensional data, over 1,800 microRNAs per sample, imbalanced classes, some stages and subtypes had fewer samples, or label leakage concerns, which we resolved in pre-processing. Our design needed to balance model accuracy with biological interpretability. Feature selection methods. Feature selection is a crucial step in our project because it reduces dimensionality and removes noise, improving classification accuracy. By focusing on the most relevant microRNAs, we enhance lung cancer detection while making models more efficient. We used multiple selection methods, each with a unique approach. Full change identifies microRNAs with the most significant expression differences. Chi-squared measures statistical dependence between features and cancer labels. Lasso applies regularization to shrink less important features weights to 0. Information gain evaluates how much a feature reduces uncertainty. Recursive feature elimination iteratively removes the least important features. NCA, Neighborhood Component Analysis, selects features that best preserve class structure. VTFS, a deep learning-based method, uses transformers to learn optimal features. Comparing multiple methods ensures that we capture different patterns in the data, leading to more reliable biomarker selection and stronger classification performance. Overlap across feature selection methods. This slide visualizes our feature selection results. The bar graph shows the number of microRNAs selected by each method, highlighting their individual contributions. The Venn diagrams illustrate the overlap across methods, showing shared and unique biomarkers. By comparing multiple approaches, we ensure a more reliable selection, capturing the most critical microRNA for lung cancer classification while reducing redundancy and noise.
Now let's talk about classification methods. Why classifications? We want to predict patient diagnosis, cancer stage, and subtype based on microRNA expression. And also classification trains machine learning models to learn patterns from labeled data. We use 3 different methods: support vector machines, or SVMs, random forest, and SAINT. Why compare multiple models? Well, each classifier may perform better on certain tasks, and the model diversity is going to help evaluate generalization and performance trade-offs. Support vector machines, or SVMs, are powerful classifiers that find the best boundary, called hyperplane, between 2 classes. So the goal is to maximize the margin, which is the distance between this hyperplane and the closest data points, called support vectors. This helps the model generalize better and reduce overfitting. In our case, SVMs are used to classify lung cancer diagnosis, stage, and subtype. In this demo, we show how we built the SVM model for lung cancer diagnosis. First, we selected the top 10 microRNAs from 6 feature selection methods. That gave us 56 unique features, down from over 1800, much easier for the model to work with. We cleaned the dataset and turned the labels into just two: cancer, 1, healthy, 0. Then we standardized the data so that all features are on the same scale. Next, we split into 80% training and 20% testing, making sure the ratio of cancer to healthy samples stayed the same. Then we trained the SVM using grid search CV to find the best C and gamma values. We use an RBF kernel because it works well for complex patterns like gene expression. And finally, we saved the model and results, including the most important features for future evaluation. Here we show how the support vector machine performed across all three tasks. SVM nailed the diagnosis tasks with 99% accuracy, catching all cancer cases, but it missed both healthy ones due to class imbalance. For stage, accuracy dropped to 52% with most predictions going to stage 1. Subtype accuracy was 49% with better performance on adenocarcinoma than squamous. Bottom line, SVM is great for binary diagnosis, but struggles more with multi-class tasks. Random forest is a powerful ensemble learning method that builds multiple decision trees on different parts of the data, and each tree makes a prediction. The final classification is decided by majority voting across all the trees. This approach reduces overfitting and improves accuracy, especially when working with high-dimensional microRNA data like ours. The idea is to apply this to the same diagnosis, stage, and subtype task as we did for the SVM. In this demo, we show how we train our random forest model for lung cancer classification. We started with 59 selected microRNAs pulled from multiple feature selection methods. This helps reduce the data size and focus on the most informative features. The dataset was cleaned, and then we added a binary diagnosis label, cancer or healthy, and then we standardized the features so they're all on the same scale. We used an 80-20 train test split, keeping the class ratio balanced across both sets. To find the best settings, we ran grid search CV over a range of values like the number of trees and estimators, max depth, and other tuning parameters. After training, we saved the best model and pulled out the top features based on importance scores for further analysis. Here we have the performance of the random forest model across all three tasks. For diagnosis, it reached 99% accuracy with perfect sensitivity. It means it detected all cancer cases. However, there were very few healthy samples, which affected its ability to correctly identify them. For stage classification, the model scored around 52% accuracy, which is reasonable given the complexity of 5 stage categories. It performed best on stage 1 but struggled with the others. For subtype prediction, it achieved 52% accuracy, doing slightly better on adenocarcinoma, the most common subtype.
This is a quick overview on one of the transformer methods used in the project. This is the VTFS architecture developed by researchers at Arizona State and Princeton. Essentially, it transforms the tabular features into tokens, then constructs an embedding space using attention heads and a variational autoencoder, similar to how LLMs work. Then it searches this embedding space for optimal feature sets. So the important thing to note here is how our first run reduced the feature set from 1,881 genes to 988, so about half. This is the other transformer method used in the project, which is Saint, developed by the researchers at University of Maryland. This works similarly to the last model, except for this incorporates intersample attention, so it focuses more on leveraging the relationships between genes. And then this screenshot shows the results of a downscaled version of the model on the miRNA dataset, where it was only able to achieve about a 50% accuracy when predicting the stage.
Hello everyone, my name's Arlie Peter. So this slide introduces the graphical user interface GUI for multi-class cancer classification system. Our goal was to create a simple, intuitive, and efficient interface for users like researchers and clinicians. We built the GUI using Next.js, React, Tailwind CSS, and TypeScript. Next.js ensures fast performance. React provides a modular structure. Tailwind CSS helps with a clean and responsive design, and TypeScript enhances reliability by preventing errors. We focused on a minimalistic, distraction-free design to make the interface easy for all users. The file upload section is a great example, clearly labeled with the drag and drop support and manual selection option. The neutral color scheme and intuitive icons keep the experience straightforward and accessible. Simplicity is crucial in healthcare applications. By reducing complexity, we ensure users can upload data, select models, classify results quickly and accurately without unnecessary steps. In short, our GUI is designed for speed, accessibility, and ease of use. By combining modern technologies with a minimalistic approach, we create a seamless experience for everyone. Thank you.
So here's a live demo of the app hosted locally. The front end is React JavaScript and the back end is Fast API in Python. So we're going to upload a sample from the National Cancer Institute Genomic Data Commons. And we upload the text file, and then we're going to choose the model as the support vector machine. So what happened there was on the back end, it formatted and processed the data. Then it selected the features based on the highest performing feature set from the research. And then it made predictions on all three target variables. And then it outputs these predictions, and the user can choose to also download these results as a CSV. So here we can see that the model predicted the patient to be cancer positive with stage 1 adenocarcinoma.
Right, on the back end, we are using the Fast API to accept CSV or text files through the API upload endpoint, which is right here. And each file is saved locally in a directory with a unique ID for tracking. With that uploaded file, we pass it to our preprocessed data function, which then loads the input file into a pandas data frame. And with that data frame, we use our select features function to filter it to include only the top microRNA columns relevant for our classification task. All right, that reduced data frame is then fed to our predict sample function, which loads our pre-trained model and returns predictions for diagnosis, stage, and subtypes. The results are then stored in our in-memory results cache here, which I do believe is a dictionary, which enables quick retrieval and download from our API status / file ID and API / download / file ID endpoints.
Discussion and lessons learned. Now that we've shown you our design, implementation, and results, let's take a step back and discuss what we learned throughout the project. First, our key findings. We confirmed that microRNA biomarkers are highly effective in classifying lung cancer, especially when we reduce the feature set to a meaningful panel. By applying 7 different feature selection methods, we identified a 7 microRNA consensus panel that consistently improved model accuracy and reduced noise. Among the models we tested, the same transformer showed the strongest performance for stage classification, reaching nearly 60% accuracy, which is particularly important because early stage detection has the biggest impact on survival. And VTFS, our deep learning-based feature selector, proved valuable with strong micro-F1 scores, especially in imbalanced classes. We also faced some important challenges, and those led to key lessons. One of the biggest was realizing how critical feature selection is when working with high-dimensional datasets. Without proper filtering, it's very easy to overfit or get misleading results. Another major issue we encountered early on was label leakage, when labels inadvertently influence the training set due to metadata overlap. We caught this during internal testing and restructured our entire pre-processing pipeline to prevent it. This improved not just performance, but also the reliability of our final metrics. In terms of deep learning, we saw strong performance from models like Saint, but these methods are harder to interpret. So we added extra steps to track which microRNAs were contributing to each prediction, which will be critical if this ever moves into a clinical setting. Finally, teamwork was essential. With 5 contributors across multiple areas, data processing, back-end, front-end, and documentation, we learned the value of clear communication, version control, and shared ownership of our deliverables. In summary, this project gave us hands-on experience with real-world medical data, exposed us to critical challenges in machine learning pipelines, and showed us how to turn complex science into something interpretable and impactful. Pulmo ML is more than just a class project. It's a proof of concept for AI-driven diagnostic tools that could help patients and doctors worldwide.